Thanks to the Nicholson Foundation as well for inviting me to be here and talk to you. I'm going to tell you a little bit about myself. Dr. Haas introduced me as working at the NIH. I'm a medical oncologist. I did my medical training in India, moved to this country in the early 90s, spent 5 years in the lab at MD Anderson, getting a Ph.D. and trying to understand how cancer genes work. Finished my residency there, moved to the National Cancer Institute, where I trained to be a medical oncologist, and have remained there since. And my primary focus is kidney cancer. I don't do anything other than kidney cancer. I don't practice in Philadelphia, but I see a lot of patients from Philadelphia. In fact, Dr. Plumack and Haas and I share a lot of patients. I'm going to spend the next 15 to 20 minutes trying to compress around 40 years of work in the field of kidney cancer genetics, a field that's been contributed to by many, many people, leaders in the field, without whom a lot of the therapies that we have today would not be possible. So I'm going to be somewhat quick and abbreviated, but I'm available for questions and discussions after the talk and during the breaks and so on. The journey really started back in the 80s when a condition called one-hipple endowed disease, which had been previously described, became a major focus of interest for a group of surgeons at the National Cancer Institute led by Dr. Marston Linehan. While a primary focus of this group was to try and understand how to improve surgical outcomes in these patients and make these patients perform better from a surgical perspective, a few things piqued their interest. So this is a condition where you get tumors in multiple organs, as you can see, kidneys, adrenal glands, pancreas, in the brain, eyes, inner ears. Many of these tumors are multifocal, meaning kidney could have many tumors both sides. These tumors started occurring at a very, very young age, and this condition ran in families. So these factors led people to say, there must be a gene that they're passing down in the family and that's causing this. So Marston Linehan, Burton Zabar, and others at the NCI then studied these families. They studied hundreds of these families and identified areas in their in the genome or chromosomes that they thought were linked to causation of this condition. And they eventually narrowed this gene down to one area of chromosome 3, and then subsequently cloned this gene that we now know as VHL, a one-hipple endowed gene. They identified several mutations occurring in the germline of these patients, and I'll go back and explain what that means in a second, and found that these mutations inactivated the protein that this gene normally made, meaning the function of this gene was lost. Subsequently, we know that patients with a more common sporadic variety of clear cell kidney cancer, which is the kidney cancer that you see in patients with VHL, also had mutations in the same gene in the same locations, except that these mutations were not occurring in the germline, but occurring in the tumor only. Several years' worth of work led to us understanding better the function of VHL. VHL has many functions. One of those is to act as a cellular sensor for oxygen. And as our previous speakers mentioned, the Nobel Prize for Medicine and Physiology this year was given to investigators, partly in recognition of their work in understanding the hypoxia response pathway better. Regardless, when you don't have active VHL in these cells, what the cells believe is that there is no oxygen. So they're trying desperately to bring in all kinds of things and to grow faster. And that leads to dysregulated growth and kidney cancer, as well as other cancers. Understanding this pathway has been critical to developing many of the therapies that you've been on or are familiar with. Most of the targeted therapies owe their origin in one form or another to us understanding the VHL pathway. In the VHL gene was identified in 1993. In the next 25 years or so, we began to understand that VHL just tells part of the story. VHL is the gene for clear cell kidney cancer, which is the first block that you see there. But we've also realized that only 75 to 80% of all kidney cancer is clear cell. So the rest are an assortment of different cancers. Some are papillary tumors, 15 to 20%. Chromophobe around 5%, and then rarer varieties that you see listed here. And as time has gone by, we've understood these tumors to be different, not just at a histologic level, but we also understand the patients with these different tumors behave differently clinically, and that there are different genes driving these different cancers. So I'm going to, before I go further and tell you what these genes mean and from a practical perspective, how they're helpful in making decisions, I'm going to clarify some of the terms I used earlier. I talked about germline and somatic mutations. What do they mean? Germline mutations are those that are identified generally by looking at your blood or your buccal mucosa and other cells, because these changes are present in all cells in your body. And you inherit them, and you have the ability to pass them on to your progeny. And usually when you see the presence of a germline mutation known to cause kidney cancer, it predisposes you to that kidney cancer. Somatic alterations, on the other hand, can sometimes affect the same genes that you see altered in patients with these inherited forms of kidney cancer, but these occur only in the tumor. So they start in the tumor and don't occur in other parts of the body. They can't be transmitted from one generation to the next. Germline testing is fairly standard today for people that we believe are at high risk for having an inherited form of kidney cancer. So who should undergo germline testing? There are no clear consensus guidelines, but several groups, such as the American College of Medical Genetics, have attempted to put out some guidelines. And when we look at patients, there are a few factors we look at that tell us, maybe we should be talking to this person or this family a little bit more about the utility of genetic testing and what's involved and what the consequences are of getting that data. The factors we consider are the presence of bilateral multifocal tumors, meaning tumors are forming in multiple areas. Young age of diagnosis. Usually kidney cancer occurs in the 50s, 60s, and beyond. Dr. Uzo mentioned earlier that to get kidney cancer, you need several different genetic events to occur before you actually start making the cancer. Some people, unfortunately, are born with one or more of these genetic defects. And so they take a shorter time to form these cancers and acquire additional mutations that would then drive their cancer. So typically, we see patients with familial kidney cancers having their cancer diagnosed at a very young age, less than 50, but I've seen patients in their 20s and even in their teens. The histology is often a very, very critical indicator of who may be at risk. There are certain histologies. There's some forms of type 1 papillary that run in families. Type 2 papillary histology, especially what we call HLRCC-like histology, which is a term that's now recognized more broadly. Patients with collecting duct, tubulopapillary, all these patients are more likely to have a germline alteration in one gene or another. And so they are often prioritized for genetic counseling and testing. We often see, because these mutations occur in every cell of the body, that other parts of the body beyond the kidney can be affected as well. For instance, if we see small fleshy tumors called leiomyomas in patients who also have kidney cancer and or have a family history, we think of a condition called HLRCC, and we test them for that condition. So those other manifestations beyond kidney cancer are telltale signs that there may be a genetic issue at play here. A strong family history. I mean, if you've had your father, grandfather, multiple members with kidney cancer, that also interests us because that's more likely to lead to a diagnosis of an inherited kidney cancer condition. But regardless, when there's any suspicion that there may be a patient that's predisposed to heritable kidney cancer syndrome, they should be seen by somebody who can counsel them appropriately. It can be a genetic counselor, it can be physicians who are experienced in this field. But that's a very critical piece of evaluating a patient for an inherited kidney cancer syndrome. A lot of patients ask me, so I have a germline mutation in a cancer gene that we know causes kidney cancer. So will I get cancer? For sure. And the answer is no, you're not assured of getting cancer. And what it depends on is multiple factors, including the gene that's affected. By way of example, I'm going to give you two genes. One's called a MET gene, which causes type 1 papillary kidney cancer. Almost all affected individuals, especially with some MET mutations, if they live long enough, will develop papillary type 1 tumors. Patients with fumarate hydratase mutations, which causes a different kind of kidney cancer, type 2 papillary kidney cancer, on the other hand, have a 15 to 30% lifetime risk of developing cancer. Because there's no reliable way of predicting who will and who won't get cancer, if we know you have a predisposition, we recommend lifelong screening to look for the onset of new cancer. So how does knowing what genes are abnormal in a cancer help? Obviously, it helps establish a diagnosis. If you establish a diagnosis and know what gene is aberrant, you can then identify family members at risk by testing them. And once you know they have the gene, you can start screening them early. You screen them early, you pick up these lesions early, remove them before they can spread. But we also know that these genetic alterations can help dictate choice of treatment. And there's some data now coming out suggesting that they may serve as prognostic markers in some cancers. I'm going to spend the next few minutes giving a couple of examples of how knowing what genes driving your cancer can dictate treatment. I'm going to use papillary kidney cancer as an example of how we can go from identifying a gene to devising a treatment. Dr. Vivek Narayan is going to be spending a little bit more time talking about options in papillary and other non-clear cell kidney cancers. I'm just going to give you a couple of examples of the work we at the NIH and others have done that will illustrate the point I wish to make. At a histologic level, when you look at these tumors under a microscope, papillary kidney cancer can be broadly divided into type 1 and type 2 or non-type 1 kidney cancer. The story, however, is a lot more complex when you look at the genetic makeup of these tumors. This is a study where investigators around the country looked at over 160 papillary tumors and did extensive genetic analysis on these tumors and found that you could divide papillary tumors based on a genetic signature into 4 groups. And these 4 groups had not only distinct genetic signatures, but also had different prognosis. So some patients did well, and those are represented in the blue and the light green line, while other patients did okay, and some patients did really, really poorly. As part of this project, we also identified a number of genes that we think may be at play in these. Not all tumors have the same genes. The genes we understand best are a gene called MET, in type 1 papillary kidney cancer. I alluded to this a few minutes ago, and a gene called fumarate hydratase in one variant of type 2 papillary kidney cancer. A lot of what we understand about the role of the MET gene in kidney cancer comes from studying a group of patients with something called hereditary papillary renal cancer. It's a relatively infrequent syndrome, runs in families, and as the name implies, patients who are affected get papillary kidney cancer, specifically type 1 papillary kidney cancer. Affected individuals have multiple tumors, hundreds sometimes, affecting both kidneys. And the histology, as I said earlier, is uniformly type 1 papillary. What happens in these patients is in their germline, they're born with an abnormal MET gene. So MET is an important gene that regulates many important cellular functions, but these people have a mutation in MET that activates the gene and makes the cells go haywire because this gene isn't under control. Often what happens is you get multiple copies of this abnormal gene, further revving up the system and making these tumors grow. But this is a pretty rare condition. Do you see MET mutations in patients who don't have this familial conditions, in patients with the so-called sporadic variant of type 1 papillary kidney cancer? We do, but not as frequently as you would with the familial form. Approximately 10% of patients with sporadic papillary kidney cancer have mutations of the same kind that you see in these families. And another 5 to 7% have other changes that we think may also activate the pathway. What a lot of people do have is extra copies of chromosome 7. Now, chromosome 7 is where MET sits. It's where the gene for the protein that activates MET sits, called HGF. And so people have said, yeah, I know if you have more chromosome 7, maybe you're activating that pathway as well. And that's a relatively common phenomenon in kidney cancer, especially type 1 kidney cancer, occurring in 80% of patients. To try and see if inhibiting this MET pathway, which we think is driving papillary kidney cancer, at least in a subgroup of patients, is feasible in the clinic, we undertook a study several years ago of a drug called ferritinib. It's a MET inhibitor as well as a VEGF inhibitor, like Sutent and other common drugs that you're familiar with. And we took 74 patients and treated them with a couple of different dosing strategies with this ferritinib. And what we found was the results were pretty disappointing. Only 10 of 74 patients had meaningful clinical benefit with tumor regression. So that's only 13% or so. What we then did was look to see how many of these patients had MET mutations and whether that impacted on outcome. Of the 67 patients on whom we had samples, we found 10 patients who had germline MET mutations. 5 of those patients, 50%, half of them, actually had a very significant response. And the other half were heading that way, but had to be taken off for toxicity associated with the drug and didn't quite achieve the tumor regression we might have expected had they continued. But if you look at patients with wild-type MET, so they didn't have this mutation, only 10% of patients responded. So this is telling you that it's a small study, but maybe having a MET mutation gives you an avenue of treatment that may not benefit other patients with papillary kidney cancer. This is an example of a patient we treated. A patient who was initially diagnosed in the 1990s, had multiple surgeries on his left kidney and eventually had the left kidney removed, has had multiple surgeries on his right kidney and had maybe a quarter of his renal function or kidney mass remaining at the time we saw him with a 3.4 centimeter mass. And the surgeons were saying, we should take this out, otherwise he has a high risk of this cancer spreading. So we put him on this drug. And within a couple of cycles, we saw that the tumor started shrinking. We kept him on drug for 4 years or so, and his tumor went from something like 3.4 centimeters to 1.4 centimeters. Didn't get rid of it fully, but significantly shrank it to a point where the surgeon said, we don't have to operate on this now. So he was taken off drug eventually and then stayed off drug for many years before needing surgery. So his clock was reset by several years because we had a drug that could target a specific genetic abnormality in his tumor. This is just to show you that if we took patients who had the MET mutation, and we had 10 of them, and they had 37 different tumors, and every single tumor that we followed on this study in patients with the germline MET mutations showed some degree of shrinkage. Again, telling you that MET mutations may predict for sensitivity to a drug that inhibits this pathway. 2 further studies, one a larger biomarker-based study of the drug savolitinib, and a smaller study in type 1 papillary kidney cancer with crizotinib, also looked at the efficacy of MET inhibitors in this setting. And we have an ongoing trial with another selective MET inhibitor called INC280. And very briefly, what these trials show is that if you have a MET mutation, you're more likely to respond than if you don't to these MET inhibitors. I'm going to very quickly switch to another inherited form of kidney cancer called hereditary leiomyomatosis and renal cell cancer, or HLRCC. This presents clinically with cutaneous leiomyomas in affected women, multiple uterine fibroids, often at a very young age. People have had hysterectomies at 20 in their 20s because of symptomatic uterine leiomyomas or fibroids, and a very aggressive type of type 2 papillary kidney cancer. This condition is not really amenable to treatments that normally work in clear cell or other forms of kidney cancer. When you look at patients in the genetic analysis, the TCGA analysis I showed you, they segregate with patients who have the worst prognosis of all patients with papillary kidney cancer. A lot of work went into trying to identify what really drives this cancer, and we found that patients with this condition have an abnormality that inactivates an important gene required for glucose metabolism called fumarate hydratase. I won't go into the details, but what this means is that these cells require a lot of glucose because they don't process glucose efficiently. They also activate the same hypoxia pathway that I spoke about earlier in VHL patients, and activate a number of genes that we think keep the need for this glucose satisfied, as well as doing many other things that cause these tumors to grow. We used a combination of 2 drugs, bevacizumab or Avastin and erlotinib or Tarceva, the latter being a drug that's used in lung cancer and has had a brief sojourn in patients with clear cell kidney cancer, but didn't really come into widespread use. So we used a combination of these two drugs to study mostly patients with HLRCC, but also used another independent cohort with patients who had sporadic papillary kidney cancer, because we know there are some similarities between the familial HLRCC patients and the sporadic patients, and we thought some of those patients may benefit as well. When this analysis was done, 20 patients with HLRCC and 21 with sporadic kidney cancer were enrolled. Most of these patients had undergone multiple prior therapies, including drugs like Sutent, Afinitor, and other agents. And when we treated them with this, we find that patients who had the mutation, that gene abnormality in fumarate heritase, with fairly predictable frequency, over 2/3 of the patients had significant tumor shrinkage. Most of the patients who didn't have tumor shrinkage had prolonged, often prolonged stable disease, meaning the tumor that was growing very fast before we started the therapy stopped growing or grew much slower, such that we could keep them on this for years. Patients with sporadic papillary kidney cancer also responded to some extent, but not to the same extent or with the same frequency as patients with HLRCC. Again, telling us that there's something about this pathway that sensitizes these patients to this drug. This is an example of a patient we treated, a patient who was sent to us being told that we don't think you really have any reasonable options, but you can go and see these guys at the NIH. You have a liver full of tumors, and I show you a representative image of 3 or 4 tumors in the liver on the left. Following a few cycles of therapy, these tumors started shrinking, and this is an image 2 years on therapy where you see very, very little residual tumor. Patient stayed on therapy for close to 4 years and then went on to get another treatment and is now disease-free. I'm going to stop there in the interest of time. I hope I've convinced you that it's very important to know what the genetic makeup of your tumor is for a variety of reasons. I went into some, but there are many more. And like I said, I'm happy to discuss this with you after the talk and during the breaks. Thank you for your attention.